Hey guys, I hope you're all well. So this is the section that will be for test 5, your last test. Woohoo! And we're going to start with chapter 18 on regulation of gene expression. And obviously, all organisms, whether they're prokaryotes or eukaryotes, have the need to regulate the expression of their genes because they don't have unlimited raw material and energy. And so they're going to interpret environmental conditions and make what is necessary. In multicellular eukaryotes, gene expression also is going to be used to regulate development. And this means that we're going to take the resources that we've got and differentiate some of the regular cells into different cell types so that they can do specific jobs. There are many roles for regulation using RNA molecules, which is very interesting, I think, given the fact that we think RNA evolved first before DNA. Bacteria respond to the environment very, very specifically by controlling only the gene products that are needed. And that's interesting because they are unicellular, they're in very close contact with the environment, so they are able to conserve their resources quite well. They regulate their enzyme production by feedback inhibition or by gene regulation. Now, this is very important because we're going to be seeing situations where, particularly with this feedback inhibition, remember that as you accumulate your final product, that final product itself acts as a feedback system which will serve to turn off the whole metabolic pathway so that we don't waste materials and energy. One mechanism that they use to control gene expression is what we call the operon model. Okay, so look at slide 6, okay? And this is an example of feedback inhibition. The production of the final metabolic product, tryptophan, provides inhibition feedback to the first step of the pathway. It's also used for regulation of enzyme production. Now, the operon is an entire stretch of DNA that's involved. It includes an operator, a promoter, and the genes that are controlled. Functionally related genes can actually be controlled in a couple of ways by one on-off switch. Now, this is important because it would be ridiculous if you were getting ready to run a metabolic pathway to have to turn the genes on separately. You know, one gene, one enzyme. What if you needed 12 enzymes in the pathway as it progressed? Then you'd have to turn them all on separately. That would be stupid. So if you are related functionally, like you're part of a metabolic pathway, we can just flip the switch and there you go. Everybody's turned on. That on-off switch is called the operator. Now remember, in the operon, you have the operator, the promoter, and the genes that are controlled. The operator is within the promoter. So there are proteins also that can switch the system off because, again, we're very concerned about wasting raw materials and energy. And these proteins are called repressors. These prevent transcription. So you want to stop something from acting, keep the protein enzymes from being produced at all. So they prevent transcription by binding to the operator, and they don't allow RNA polymerase to bind at all. So you cannot run that process. There are what we call regulatory genes that produce the repressor, and they can be, the repressor itself can be in an active or an inactive form. There are things called co-repressors that cooperate with the repressor to turn the operon off. For example, in E. coli, they can produce the amino acid tryptophan if it's lacking in their environment. So the trip operon is on all the time. Okay, now wait a minute. Let's back up and think about this. So we said that if the amino acid tryptophan is lacking, we can produce it. So isn't it interesting that it's not always lacking, but the trip operon is already on all the time, ready to go to work. And the genes can be transcribed to make tryptophan. Now, what if there is enough tryptophan in the environment, in the media that we're growing on? Well, the tryptophan itself binds to the trip repressor protein and turns the operon off. And what a neat signal that is, to let tryptophan itself be the signal, hello, we got enough, okay? So the trip repressor protein is only active if the co-repressor tryptophan is present. So the trip operon gets turned off or repressed if the tryptophan levels are high, because we don't need it. So study slide 11 and it'll help with that a little. Okay. Repressible operons are ones that are usually turned on, and you turn off transcription by binding a repressor to the operator. So a great example of a repressible operon is like the trip operon we just talked about. An inducible operon is one that is usually turned off. And you need, the inducer is actually going to inactivate the repressor and turn on transcription. Now, a great example of that one is the lac operon. Now, for the lac operon, we're going to be coding enzymes that are involved in the metabolism of the carbohydrate lactose. If the lac repressor is alone, it's in its active form, and it turns the lac operon off. If you have an inducer present, and it's present with the lac repressor, that will serve to turn the lac operon on. Okay? So study those slides, and I think that that will help. Inducible enzymes, like those involved in the lac operon, are usually involved in catabolic pathways. Those are breakdown pathways. And their synthesis comes from a chemical signal. Repressible enzymes usually involve anabolic pathways, which are pathways building materials, and their synthesis gets repressed by high levels of the end product. So remember, repressible are on all the time. The trip and the lac operons can be turned off by active form repressors, and this is called negative control of genes. With positive gene regulation, we use what are called stimulatory proteins. And a great example is cyclic AMP receptor protein. It is an activator of transcription. So, for example, what if I told you, which is true, that E. coli really prefer glucose as a food source? If it is scarce, then cyclic AMP receptor protein gets activated by binding with cyclic AMP. The activated cyclic AMP receptor protein attaches to the lac operon promoter. It increases the affinity of RNA polymerase and increases transcription. So if glucose is scarce, then the bacteria can automatically go to the lac operon and develop the materials that it needs to use lactose as a food source. When glucose increases, the cyclic AMP receptor protein detaches from the lac operon and transcription goes back to a normal rate. The cyclic AMP receptor protein helps regulate other operons that encode enzymes used in catabolic pathways. So, for example, if we can catalyze lactose, even if we lack glucose, then we can survive the current conditions. Compounds that are present in the cell ready for use will determine which genes get switched on. All organisms must regulate which genes are expressed at any given time. We just don't have a choice. There's not that much as far as raw materials and energy. Genes are turned on and off, remember, based on signals from your external and your internal environments. And remember, in multicellular organisms, gene expression regulation is the way that we do cell specialization. All cells of an organism have basically identical genomes.